Rahim. Good afternoon, everyone. I hope you are all doing well. My name is Dr. Mohammad Khurram, lecturer and research coordinator of the engineering department. I am the moderator for today's session, and I'm excited to be here with you as we explore today's topic. First of all, I warmly welcome you all. Today we have lined up an exciting and insightful session where our speaker will be discussing the research-oriented topic related to emerging field of artificial intelligence. As we all know, AI, artificial intelligence, is transforming industries globally, and understanding its impact on our life is crucial for both businesses and individuals. So, moreover, AI is one of the main objectives of Oman's National Innovation Strategy 2040. So, this strategy aligned with Oman's ambitions to rank among the top 50 countries in the government AI readiness index. So in line of this, University of Technology Applied Sciences has taken multiple initiatives to improve the research culture and support the implementation of AI. And we are trying to provide the latest knowledge about the field of artificial intelligence. So to this end, the Research and Consultancy Department of the University of Technology Applied Sciences NISWA is launching a series of sessions focused on AI, data science, or the other like latest technologies. And we are inviting renowned researchers and academicians from national and international level. So today we are honored to have our guest speaker, Dr. Fawaz Salam Khan, with us, who will share their expertise on this important topic. So before we get started, just I want to share the few points regarding the decorum of the webinar. I request to all of you all attendees, please make sure your microphone are muted during the session to avoid any background noise. If you have any question, feel free to type them into the chat box. Our speaker will address them at the end of the session. Our session will run for approximately 1 hour, not more than that, and we will wrap up with a question answer session at the end. So please stay tuned until the end. At the end, I will also share the feedback form as well. So all you please must fill out. So now I am excited to get started and hope you find this session valuable. So without any further delay, I will hand over to Dr. Saif Al-Bimani, Head of Research and Consultancy Department, to introduce our speaker. Dr. Saif, are you
Yes, Dr. Mohammed.
here? Okay, sir. Please carry on, sir.
Thank you so much. Okay, Assalamu alaikum, everyone, and a special welcome to our guest speaker today, Dr. Fawad. Um, Good afternoon, Doctor, and thank you once again for joining us. I'm
Thank you
sure
very
it's
much.
going to be quite informative, and it's going to be a learning experience for all of us about one of the most important topics that we're going to discuss during this session today.
Yes.
Before we begin, I'd like to introduce our speaker. Dr. Fawad Salam Khan is a postdoctoral research fellow at the University of Lille, France, funded by the National Center for Scientific Research under the Talent Visa France program. Dr. Fawaz completed his PhD in electrical engineering, artificial intelligence from University Tun Hussein on Malaysia, supported by the FRGS Malaysia grant. He also holds a master's in computer science from International Islamic University, Islamabad, and a master's in computer systems from NED University, and a bachelor's in computer engineering from Sir Said University of Engineering and Technology, Pakistan. Dr. Khan has over a decade of academic and research experience, having served as an assistant professor and department chair at Air University, Islamabad. He has led and secured multiple international research grants totaling over 300,000 US dollars, including projects funded from Silicon Valley, the UK, and Malaysia. His postdoctoral research focuses on lung cancer anomaly detection using LIBS spectral data and deep learning architectures. He has published extensively in Q1 and Q2 journals, including IEEE Access, Sensors, Applied Intelligence, and CMC, covering domains like deep learning, reinforcement learning, and UAV control. He's a recipient of gold and silver medals at RISE Malaysia and a run-up in the IEEE 3MT competition. Dr. Khan has supervised numerous PhD, master's, and bachelor's theses in AI, deep learning, and biomedical engineering. His current research interests integrates machine learning, spectroscopy, and robotics, advancing intelligent systems for biomedical and autonomous applications. It's quite an accomplishment, Doctor, here. So thank you so much once again, and the floor is yours, Doctor. So please go ahead. Thank you.
Okay, first of all, thank you very much, Dr. Saf, for giving me this opportunity, and also Dr. Khurram for having this webinar for this topic, machine learning optimization with Optuna for hyperspectral lung cancer anomaly detection. So as you have already given my brief introduction, I'm just skipping this slide. So let's dive into the content. Basically, we are going to start with the issues related to the global cancer issues. Normally, in the statistical overview, is these days about 2.5 million new lung cancer cases projected globally till 2025, and about 1.8 million deaths projected annually, which is like 18.7% of all cancer deaths, which causes leading the cancer mortality worldwide. So normally the key issues regarding the lung cancer are the large stage detection reduces minimum survival cases. And the rates of survival is very minimum if there have less chances for the detection. So traditional diagnostic methods are labor intensive, which can produce subjective interruption, leads to diagnostic variability. So in the early, you can say we need some early and accurate detection methods where we are going to have some different concepts in machine learning and deep learning, which will be discussed in this presentation. So before starting the major concepts of the optimization, I'm just going to have the hyperspectral imaging, a breakthrough technology overview. Basically, it is HSI, you can say hyperspectral imaging is a technique that captures and processes the information from across the electromagnetic spectrum for each pixel in an image. You can say, unlike the human eye, which we see colors in three different, as you see in the slide, in three primary bands, you can see a red, green, and blue, and standard cameras which do into the same. A hyperspectral camera captures light in many more, which are narrow, contiguous spectral bands. So think of giving it a pixel in an image or an unique chemical fingerprints. So what HMI offers normally, it captures a data across numerous narrow bands ranging from 420 to 750 nanometers. So there can be numerous advantages like non-destructive and non-contrast. Like, for example, if you analyze an object without touching or damaging them, you can have some kind of biochemical or pathological information which can reveal both spectral information, have different kinds of combined spectral and spectral dimensions. So there can be various challenges as well. You can say said that hyperspectral camera has the system which significantly more expensive than standard cameras. So there can be multiple clinical applications you can use in lung cancer, for example, melanoma classification, or you can say cervical cancer detection, or head and neck cell carcinoma, lung tumor identification. So there can be various applications in clinical side for hyperspectral images. And these days, you can say one of the major important stream of research for lung cancer detection. So similarly, we can detect different kind of invisible reveal features from these properties from the hyperspectral data. So normally, if you can say why we are use hyperspectral imaging that can be, it is in the form of continuous spectrum, which provides both spectral and spatial information of the image features. So similarly, if we are talking about the role of machine learning in medical diagnosis, you can say, so why we can use machine learning in medical diagnosis? There can be various reasons for that. Like for example, it automates pattern recognition in high dimensional data. As if you are comparing the normal image, which is of the 2D, and the hyperspectral data is normally is a very high dimensional data. So machine learning can provide an easy way for the detection and prediction for these type of research or research areas. So we can extract some kind of features from which can be observed from the human eye, or which can be missed from the human eye. Sometimes it cannot be find easily to get some features manually, and the machine learning algorithms will help out to provide an objective and reproducible results, which can scales up the large data set efficiently. So there can be various challenges, like for example, if you are saying high quality data, if you don't have high quality data, that means the machine learning algorithm cannot perform it very efficiently. So it requires high quality labeled data sets, which have ethical regulations, limit data availability. Normally, if you're talking about in France or in Europe, you cannot get any data easily from the hospital or any kind of such organization without the permission and without having a proper NDA signed with the doctors and the hospital. So this can be a very big challenge for us for getting these type of data sets from the patient. So another more important challenge is the annotation, which is normally it's very difficult, because we need a qualified doctor for that, for the annotations, and very high quality impacts models performance are required for that, and they have multiple, you can say, annotation, like for example M6 4000 devices here, so get the annotation done. So for the hyperparameter optimization, which is another a very crucial, you can say a crucial site for the optimal results, which can be implemented in my research. So I'll discuss step by step. So if we are talking about hyperspectral HMI with traditional RGB imaging, we need to understand that this can be very important. First, we understand the basic concept and the basic difference between RG images versus the hyperspectral image. So instead of just 3 values of RGB for a pixel, you just hundreds of values representing the light intensity at each specific, you can say, wavelength. So hyperspectral camera are normally used to capture hundred of narrow contiguous spectral bands, for example, from 400 millimeters to 2500 millimeters, that creates a complete detailed spectrum for each pixel. So for example, if you are having a basic layman difference concept between RGB imaging, so RGB imaging consists of 3 spectral bands, normally red, green, blue, which has a limited to visible spectrum, captures basic morphological features and lack of biochemical information. As you see in the figure, there is a difference between 3 colors, and you can see in hyperspectral there is a cube which contains 3 different values. Although the RGB image has also 3 axes, X, Y, Z, X, Y, and lambda. Here also an X, Y, Z, but the hyperspectral image is consist of more spectral frequencies, and which makes the image in the form of 3D, or you can say in a cube. So hyperspectral imaging normally have 67 plus spectral bands, which have rich spectral signatures, capture molecular composition, detect subtitle tissue changes. So the major advantage for having hyperspectral image is basically each pixel contains complete spectral profile, enabling differentiation based on chemical composition rather than just an appearance. So in the RGB image, all the 3 colors like red, blue, or green have a different kind of visible spectrum as compared to the hyperspectral imaging. All the combinations are in the form of, you can say, the complete wavelength, which contains the value from 400 nanometers to 2500 nanometers. So hyperspectral, what are the characteristics why we are using these hyperspectral data? For example, so the data cube which you have seen in the earlier slides are normally consist of 3088 into 2064 into 64 into 67, means width, height, and spectral bands. Each pixel 67 dimensional spectral data, which is very big data set. Total information is about 6.4 million pixels into 67 bands. That each band contains, means 64 million pixels. So there can be an spectral signatures, like for example, if you say cell nucleus, this is the basic combination of the data set for that cube, which consists of a cell nucleus, cytoplasm, and background tissue. So the what we are having the interest is cell nucleus, which consists of a distinct spectral curve, and a cytoplasm, which have different spectral patterns, which have different ranges of wavelengths, and then background tissues, separate signatures like tumor or non-tumor. Like for example, for the prediction and the classification, we have to find out that this cube is consists of a tumor or a new tumor position. So for these are type of spectral signature, we must notify that for the prediction we have some kind of challenges which have, like for example, high dimensionality increase the computational complexity and storage requirement, which is majorly the most difficult phase, because if you don't have a more computational power, for example, I have multiple clusters of GPUs here in France for resolving all these kind of issues regarding to the dimensionality, by handling TensorFlow or Keras or TensorRT. Different kind of applications are available in the GPU, running with the CUDA. So data processing pipeline is one of the most important concept here for solving any kind of a machine learning or a deep learning issue. For example, we need to first we need to understand the data cube generation, as I told you earlier, that that it always stacks in a single band image into multiple band format. So the band format means the wavelengths of each spectral range is different from one to another. For example, we have multiple cubes which contains different wavelengths. So the raw data must be stored in the raw files in metadata, which can be a CSV or JSON or anything like that. And we have a data collaboration. For example, if you see, there is a formula. The output of an hyperspectral scan is not a simple 2D picture. It's just a three-dimensional data set called an hypercube or a data cube. So if we are talking about the three dimensions, for example, X, Y, these forms of a spectral dimension, just like a normal image, they define the picture length and the width easily. And Z-axis, which is most important, you can see a lambda. This is a spectral dimension for a single XY coordinates pixel in the image. There is, you can say, full detailed spectrum. For example, if you are talking about why we are using this, to understand the purpose, we need to reduce the high frequency noise in a single in a signal, you can say, like a spectrum, while preserving in a essential shape and features like peak, positions, and width. It is convolution-based methods that performs local polynomial regression, where we can reduce noise filtering and normalization by applying sophisticated Gole smoothing with window size 5 and polynomial 2. Standard normal variate SNV normalization, which is very important here, in which we have subtract the mean of each spectrum or spectra to another spectra and divide it by its derivative. So it will reduce the noise and the normalize the data set, which contains the most important information of each spectra inside that. So we use also a sliding window concept, a window of 5 data point slides throughout the spectrum, 1 point at a time, or you can say a local polynomial fit for each window position with second level of polynomial, and have a dimensionality reduction procedure where we use PCA, principal component analysis, which are normally used to remove the redundant information, improves the efficiency. So normally if you see in the spectra, there can be multiple values of the spectra, or peak contains a redundant information which is unuseful. So we need to reduce it or remove it by applying a principal component analysis, PCA. So there can be classification algorithms, which we have already two type of machine learning and deep learning algorithms. One is random forest machine learning ensembles that, like we have decision trees for the random forest, and have support vector machines for like kernel based classification and stock generalization. And in the deep learning, we have applied 2D CNN extract spectral feature, and 3D CNN captures both spectral and spectral features. And in my concept for the research, we have applied an hybrid SN attention model, which combines 3D and 2D CNN with attention mechanism. So the methodology is based on this, you can say in the flow chart. Like for example, we have a lung data set, then we apply a different kind of class balancing to contains the data set for the different efficiency, and then apply random forest, ADA boost, X boost for the classifier, then apply Optuna based optimization, we'll discuss in the next slides. Then about apply model evaluation. After that, in the last stage, I have applied lung cancer prediction. So traditional optimizer versus for example, here we can say all you know that grid search is a traditional optimizer which already used in machine learning and deep learning. It's basically a widely used hyperparameter optimization technique which operates a brute force principle. So it involves defining a grid of you can say hyperparameter values, and majorly searching through all possible combinations, which will take majorly more time. And grid search evaluate each combination independently, which is very difficult for a processor, or you can say have a different specs of containing training and evaluating models for each parameters. Although grid search guarantees that all the combinations will be explored. So you can say if we have the concept of exploration and exploitation, grid search is more towards on the exploration, as it's find out all the combination possibilities, and it can be computationally expensive for that, especially with a large number of hyperparameters. In the case of hyperspectral imaging, there are multiple many combinations are available. So the large number of hyperparameters or the wide range of values are very difficult to find to optimize or tune. Additionally, grid search lacks adaptability, you can say, to handle the non-linear relationship between hyperparameters, making it less efficient. We just define the grid of hyperparameters values, which evaluates all the combinations, select configuration, and best validation performance. So there can be advantages and disadvantages for that. Simple to implement and understand. Grid search is very easy. Guarantees exploration. As I say earlier, that all the combinations are possibly explored in each of the search spaces. Results are easily interpretable. Then there can be multiple disadvantages as well. Because of the exploration, it is computationally expensive, inefficient for high dimensional spaces, treat all regions equally. Means that, for example, if we use PCA for, if we don't use PCA for removing and apply grid search, and it will not give a better results, because the space will be very large. So for example, if we have hyperparameters, 5 hyperparameters, and which have each of 10 values, so there can be 100,000 combinations to evaluate, which is very high. So these are only for the 5 hyperparameters. For example, in hyperspectral imaging, there are multiple hyperparameters. For example, you can say 30, 40, 50 hyperparameters, which have different values. So it will make a very big combination to evaluate. So now introducing a very important Optuna algorithm, which is another very new and good optimizer. So Optuna is a modern hyperparameter optimization framework, a different approach. It leverages techniques from Bayesian optimization and stochastic optimization to intelligently explore the hyperparameter search space. So Optuna employs an algorithm called tree structured Parison estimator TPE, which models the relationship between hyperparameters for evaluating the combination and building a probabilistic models. So Optuna focus in such on the most promising regions of the hyperparameters such spaces, leading faster convergence and improve efficiency. So if you say what are the key innovations for the Optuna, it can intelligent, adaptive search strategy, learn from previous evaluation, dynamically adjust search direction, prunes unpromisingly trial early. So there can be the integration is quite much available in different scenarios. I have applied in scikit-learn, TensorFlow, PyTorch, and other ML frameworks. So there can be a major difference between grid search and Optima. You can see grid search is well suited for scenarios with the search space is small and the number of hyperparameters is limited. It can be provide reliable results while exploration is feasible and computational resources are not concerned. So Optuna shines in scenarios where search space is large and numbers of hyperparameter is normally sustainable. And it efficiently navigates complex optimization landscape, making it ideal for deep learning and machine learning model, which are computationally demanding more tasks. So if you say that the search strategy between two is like, for example, in grid search, it use a systematic strategy. In Optima, it is intelligent and adaptive. You can say in efficiency, like for example, if you see O in n n power k, where n is the values and hk is the hyperparameter. Similarly, Optima give more significantly fast convergence. So adaptability, resource usage, best cases. So I have applied research and Optima and find a major difference between both these algorithms, that Optima performs majorly a very good results in hyperspectral scenarios. So in medical imaging, why we use Optuna for medical imaging? It is computational efficient. Then complex optimization landscape, there can be resource constraint, and the customization can be one problem. So in medical data sets are very large, Optuna reduce training times efficiently. So normally, like for example, in my scenario, it will take not more than 3 to 4 hours for 100 to 200 epochs, depending upon the data sets and a number of parameters which I use, like learning rate or batch size, or you can say different kind of parameters. So hyperspectral data has high dimensionality, nonlinear relationship between hyperparameters. Optuna handles complex effectively. So there can be medical researches which have different computational budgets, and Optuna, if you apply Optuna in that scenario, it can maximize the performance as per the hardware, as per the evaluation, as per the different matrices. So we have defined different custom search spaces which have domain specific optimization specific objective. For example, I have applied Optuna for denoising different kind of scenarios for the removal of different you can say inefficiently products inside the data set. So it can integrate medical performances matrices as well. So for the experimental setup, I have hardware configuration scenario with Supermicro 7049 GPTRE workstations, 2 into Intel Xeon Gold 5118 CPUs with 4 NVIDIA GeForce 2080 Ti GPUs and Linux operating systems. So for example, the most important in this slide is the patch generation. So having all these information, I just give the concept of a patch. So patch is normally a sub-region or a block extracted, you can say, from a larger medical imaging, for example, MRI, CT scan, X-ray, or histopathology slides. For example, here I have taken 25 cross in 25, but in the if I say in the form of MRI, 512 into 512 MRI slice may be divided into a patch of size of 64 or 128, 128. So these patches can overlap or be non-overlapping depending upon the application which we have. So in my scenario, I haven't used non-overlapping scenarios. So it will produce memory efficiency, data augmentation possibilities, focus on local features and improve the efficiency. So in this scenario, the training parameters are Adam. Optimizer used is Adam. Initial learning rate is 5 exponential minus 5. Then batch size is 256. I have tried with different batch sizes like 32, 64. These are the best 256. Epochs starts from 50, 100, and 200. Training test is split into 8:1. So lung cell carcinoma LUSA data sets I have used here. So it's a key laboratory respiratory, which 10 pathological slides. So annotation was full label data sets with non-cell, non-tumor cell, tumor cell, and the background, which is normally the segmentation block depending upon the doctor which have already annotated here. So cell level precision through k-mean refinement varied by an experienced pathologist. So we have the composition of the data set with like 65 hyperspectral image. Each image dimensions is 3088 into 2064 pixels with 67 spectral bands, 57 after noise removal. The wavelength here we have taken is 420 to 750 nanometers. So the overall scheme of a lung tumor classification from hyperspectral pathology starts from acquisition of the data sets, then have prepared a sample preparation, then applied, then we get the raw data collection. After that, there is a huge line of pre-processing step like calibrations, then noise filtering, normalization, dimensionality reduction using PCA, then label preparation. After that, the hyperspectral image data sets has data cubes and labels. So the data cubes and labels are then having the classification model like RF, support vector machine, and CNN. After that, we have an analysis, or you can say an evaluation, which have different values like sensitivity, accuracy, dice, and specificity. So the model architecture which we have applied here is a 3D convolution layers which used to extract spectral features, and have convolution layers which is focused on the spectral, normally spectral data feature extraction, refined feature maps from 3D processing. Then we have attention model, like for example here we use channel attention, and which emphasis with the important spectral features like highlights relevant regions, improve feature dimensionality. And lastly, the model consists of batch normalization, which stabilize the training, accelerate convergence, and improve the generalization. This is the model architecture. So Optuna integration with this model is like we have different 5 steps here. So first we define the objective function in which hyperparameter configuration is defined. Then for the output, we have validation, accuracy, and loss. In the second step, specify the search space learning, for example, 1 exponential minus 5 till 1 exponential 3. Then number of layers, which is starts from 2 to 6. Filter size, dropout dropout rates, etc. Then the third step is differentiation, which can produce maximize, minimize, or TPE, random grid optimization techniques. In the fourth step, we have applied the optimization, which can evaluate the configuration and prunes the upcoming promising trials. And the last step, extract the best parameters, refine the model, and then apply it on the major data sets. So the optimization strategy is basically we have a random forest with have n estimators, number of trees, maximum depths, trees depth limits, and minimum samples for centering. Similarly, we have SVM, like regularization parameters. Kernel have RBG, polynomial Gaussians, then have learning rate, number of 2D, 3D convolved layers, filter size, which is starts from 3, 4, 5, dropout rates, attention model parameters, and the optimization metrics, which is primary accuracy, secondary sensitivity, and dice coefficient. So this is basically a performance metrics where we have applied accuracy, which is the true positive overall correctness of the classification, true positive plus true negative, divided by all of these model definitions. And then we have recall, ability to detect tumor cells, minimize misdiagnosis, and the ability to correct, identify non-tumor classes. Then have dice coefficient, which measures the segmentation overlap to find out the backgrounds of that image from the hyperspectral data set. So these are the experimental results. As you can see, the hybrid SN attention model provides the best in all. Accuracy is higher, specificity is higher, sensitivity is to 54, and then dice is 55. So the key findings from this experimentation has given me the great attract best overall performances from the deep learning, traditional ML for tumor detection. Hybrid architecture combines spectral and spectral information. Impact of label refinement, because labels are mostly most important point here for finding out the best accuracy in terms of, you can say, course and cell labels, which is consist of course have 77.33 and cell label is 92 point, which is the best higher accuracy. Analysis is like plus 15.19% in accuracy improvement and plus 12.58 specificity improvements. So majorly, the cell level guide models to focus on spectral characteristic of individual cell rather than tissue level differences. So if you are talking about the hyperspectral imaging, so spectral characteristics which have different levels of wavelengths in each spectra have different cubes. So each cubes have different characteristics in which we have find out this difference, like plus 15.19 and 12.58%. Now, the traditional model comparison, you can say, is like we have a traditional ML have struggles with tumor and non-tumor cell distinctions, whereas my model has best tumor cell identification, most stable predictions, effective use of attention mechanism, balance in both feature extractions. So clinical values provides about you can say for the pathologist, it will increase the sensitivity up to 50%, which is very much good for the pathologist and other doctors in hospital, which will give a better results and getting more better accuracy for the detection of lung cancer for the any patient. So key finding for the clinical operations, implementations, you can say applications, efficient annotation methods reduces labeling time from 30 minutes to 5 minutes approximately slides, but it is like it always depends upon the pathologist and the equipment available in any doctors in any hospital, you can say. It can maintains high level of accuracy, 92.5%. Then the most important which we have gathered the information is like the hyperparameter optimization produces the efficient model tuning, which can give the achieve which is used to achieve the optimal performance for the fewer evaluation. So it can be used for superior detection performances, like 54.34 sensitivity for tumor detection, which is quite good. So if we say about what are the impacts on the clinical side, so it can assist the pathologist in rapid tumor identification, reduces diagnostics time and subjective variability, provide objective reproducibility results. Like for example, if you reproduce the results with the better accuracy, so that means that it will give more best results in terms of accuracy and sensitivity for the lung cancer detection. And normally, this scenario can be implemented for the early stage cancer detection. Like for example, if you're talking about the first stage cancer, it can be more important to apply this scenario. So we can find out some limitation and challenges here. Like for example, we have only 65 hyperspectral images, a single cancer type, LUSA, may not generalize to other lung cancer subject types. So in the future, we can apply on different data sets to get more better results. Annotation method K-mean clustering requires manual verification. So this is the most important observation which we have observed, that every time the annotators are doing work and have the validation and go back to the doctor and then to validate all these information, whether the K-mean clustering, that each pixel value having a right point of extraction from the wavelength is accurate or not. So it's very difficult every time to move around. Then we have the model structure. Sometimes CNN struggles with larger range of spectral dependency. So not every time CNN giving a better results. We can apply LSTMs as well. We can apply, you can say, marginal algorithms as well. So there can be multiple deep learning algorithms, like, for example, you can say normally these days spin models, PINN is physics induced neural networks are normally used for the model architecture as well. So there can be different clinical validations as well, must require testing, and diverse patient population need integration with clinical workflow. So for the future direction, I can implement, I'm trying to implement these day vision transformers for long range of dependencies. UNet has already applied for segmentation, and it gives some kind of better results up till now. But I don't have any kind of validation till date with the doctor. So I will use hybrid transformers with CNN models. Also have applied semi-supervised learning, which can leverages unlabeled data, iterative label refinement, but there can be differences because the ranges of the frequencies or the peaks are very difficult to understand every time. So in my you can say opinion, semi-supervised learning is quite much difficult to implement. Active learning strategies can be applied. Then we have expanded applications other than these type of small cells, multi-region pathology analysis, real-time incorporation, and guidance. So uh, for my future directions, there can be interactive segmentation tools for the pathologist, which can be applied for early level of you can say lung cancer detection. So an integration with hospital information system. Again, we are talking about some of the hospitals in France, but we need some kind of approvals or NDA sign every time. So it will take time to get all these done. Then we will apply prospective clinical trials. Again, these we like, for example, we have 65 data cubes we can implement on different perspectives on the data sets. Then we have regulatory approval pathway, which will take about 1 to 2 years from this. So uh, in the last slide, I can say the conclusion of the same, that, like, for example, the Optuna advantage efficiently navigates high dimensional hyperparameter spaces, adapts to complex nonlinear relationship in deep learning models, which is like, if we say, I have the experience for getting linear and non-linear relationship in deep learning, but I find non-linear relationships are majorly best solved by the Optuna. Reduces computational overheads and maximize diagnostic performance, essential for resource-constrained medical AI research. So clinical impact, you can say, we have the reproducible tumor detection, reducing inter-observer positions, and then have rapid analysis, cell level precision, capturing suitable changing. So there can be like, for example, in my view, Optuna can perform the best efficient method in higher dimensional hyperparameter spaces and adapts nonlinear relationship. So that's all from my side. Thank you very much.
Okay, Dr. Fawad, thank you, and so nice of you. Very interesting topic you are delivered to us related to machine learning and optimization techniques. Now, uh, if anybody asks any question about the topic to ask Mr. Fawad, so you feel free to type in the chat box, or you ask verbally. Mm, just yes, if anybody have any question. Miss Sangeeta, she asked the question, I think she thanked to you, and any specific regularization method used?
Yeah, for the regularization method, I use L1 and L2 regularization for both the scenarios. For the hyperparameter tuning, I use L2 regularization.
Okay. Uh, Dear attendees, here I also shared with you the feedback form regarding this session. So all you please fill up this form before leaving. Please kindly uh, fill up this form I shared here in the chat box. Dr. Fawad, I ask one question to you.
Sure.
Uh, for any diagnosing like algorithm, why the optimization is impactful in machine learning or these related issues?
Okay, as I told in the presentation that hyperspectral imaging or normal imaging, sometimes there can be a huge number of parameters. So optimization is more necessarily to get the done to solve all these type of problems to with which have large dimensional data sets. So optimization can provide like in hyperspectral imaging or feature optimization, we can use optimization techniques.
What do you think is this Optuna can be used other the hyperspectral data optimization is possible?
Yes, yes, it's possible. I have
Mhm,
applied in 2D and 3D both data sets other than hyperspectral data.
mhm. Okay, thank you. The one more question I ask, like journal question regarding the uh, PhD and the postdoc. Mashallah, you are doing the postdoc in France, and you are working in National Center of Scientific Research. So, could you explain a little about to our attendees that how to apply those they are interested in PhD and those suppose they are interested in postdoc, so how they get the funding and how they like to get the admission in PhD or postdoc. Could you explain little about your
Yes,
experience in France
sure.
and what the procedure to get the funding, all this process?
Okay, sure. Basically, I have been selected out of 62 candidates for this position for the postdoc in anomaly detection lung cancer by CNRS. So I have applied through, there are three official sites. One is CNRS and the other one is INRIA. I can share the details later on email or something like that. So for that, they announce position regularly in few months. Right now, I think
Hmm.
3 or 4 positions are already there. So I can also
Okay.
share with these things with you guys as well. So
Uh-huh.
there can be a scenario, for example, if you are applying for particular domain, you must have some kind of research experience and some publications as well. Especially, for example, for the medical diagnosis, you must have some Q1 and Q2 papers on that as well. So
Hmm.
this can be first step. So the second step is like they can if you have been shortlisted, they will take you for the test, and this test has 3 to 4 steps normally with the supervisor or with the organization. So the test consists of specifically more specific on the field, and if you get all the tests done with good marks, you will be shortlisted for the interviews. Then the interviews consist of two portions. One is with the expert, and the other with the HR. And after
Okay.
that, if you find out if you just you can say pass the technical interview, then you will go with the HR, and then you will be selected. So there can be like, for example, 40 students around, you can say not only 40 students in this semester. in from India, from Pakistan, from Sri Lanka, and from different Asian countries. They come here this year. So I've been visiting Belgium and Netherlands last month, where I visited and gave some lectures to other universities. There are also some options available for PhD and postdocs as well. I will share all these options with you guys. Yeah.
Okay, okay, fine. Thank you. So nice of you and Dr. Fawad to give us the your precious time in your research time actually. So again, I thank you and on the behalf of Dr. Saif as well because now he left. He went for the some meeting in the university. And thank you all for the attendees taking the time to join us today. Your participation and engagement have made this session truly meaningful. So we hope you found the webinar valuable and informative. Please feel free to share your feedback. I already share the feedback form in the chat box, and also I will share in the WhatsApp